Hello friends, this is Dr. Anjani, Assistant Professor in Biotechnology. Welcome to e-learning platform. Today we'll learn about cellular basis of immunity. Cells of immunity. The cells of immune system are present as circulating cells in the blood and lymph. They are distributed to almost every organ and tissue of the animal body. Their distributions upon exposure to an external agent or pathogens are at most important in the generation of effective immune reaction. At the beginning, the immune system must respond to the entering pathogen, followed by an adaptive immune response by specific lymphocyte. Finally, the cells of adaptive immune response destroy the pathogens by the effector cells. These cells of immune system are derived from, or they are originated from the blood stem cells. They are originated from the blood stem cells, and from the blood stem cells we will find two important lineages. One is myeloid stem cell and another one is lymphoid stem cell. Under different environmental condition, these myeloid stem cells develop into RBCs and then platelets. From the myeloid stem cell we will find the formation of the granulocytes also. So these granulocytes will be having the granules in their cytoplasm, and these granulocytes are involved in the immune reactions. This include neutrophils, basophils and eosinophils. And these granulocytes, they have the lobed nucleus, that may be bilobed, multilobed nucleus, and these are called as polymorphonuclear leukocytes. These are called polymorphonuclear leukocytes. This is regarding myeloid lineage of stem cells. Apart from these cells, we also find the macrophages and monocytes. They also will be originated from the myeloid stem cell. Next comes the lymphoid stem cell, that also originated from the blood stem cells by the process of hematopoiesis. And in the lymphoid stem cell, the lymphoblast, finally they form either B lymphocyte or T lymphocyte or the natural killer cells. So B lymphocyte, T lymphocyte and natural killer cells are originated from the lymphoid stem cells, whereas the granulocytes, macrophages, monocytes are originated from the myeloid stem cells. These granulocytes and the lymphocytes, or the B lymphocyte, T lymphocyte and natural killer cells, will be considered as white blood cells. They play an important role in immune reaction. So this is the list of the cells which are involved in the immunity. The myeloid cells include the macrophages and the phagocyte, polymorphonuclear granulocytes and monocytes. And the lymphoid cells include T cells, B cells and natural killer cells. First we will see the phagocytes and macrophages. Phagocytes and macrophages, they exhibit the process of phagocytosis. They are present in virtually every tissue and organ of the body and respond instantaneously to entering pathogen. Immediately they respond to the entering pathogen. The job of the phagocytic cells involve the recruitment of the cells at the site of infection, ingestion and destruction of pathogens. So here the important processes phagocytosis will be exhibited during the removal of the pathogen or in overcoming the infection. Next we learn about neutrophils. These are granulocytes present in the bloodstream and are the first line of defense in the body. They are the first line of the defense in the body. They are the most abundant cells present in the bloodstream. They are about 12 to 15 micrometer in diameter with projections on their surface. The plasma membrane shows the extensions. These are called projections on their surface. The nucleus of the neutrophils contain 3 to 5 lobes. Hence I told you granulocytes are called as polymorphonuclear leukocyte. Polymorphonuclear leukocyte. Morphologically, when you see the external appearance of the nucleus, show the many lobes. Hence they are called polymorphonuclear cells. The cytoplasm of the neutrophils contains the granules. The cytoplasm is granular in nature and they contain or they are filled with the enzymes like lysozyme, collagenase and elastase. And these neutrophils stain with neutral dyes. Hence they are called neutrophils. And produced in the bone marrow. They are produced in the bone marrow. These neutrophils are on an average per day about 10 to the power of 11 cells or 1 to 10 to the power of 11 neutrophils will be produced in an adult per day. Per day an adult individual produces 10 to the power of 11 neutrophils. Usually neutrophils are recruited at the site of infection immediately following the invasion of the foreign substance. If not, they undergo apoptosis and get cleared from the circulation. The other two are the basophils and the acidophils. Next we learn about macrophages. Macrophages are actively involved in phagocytosis. They arise from the bone marrow stem cells or circulating monocyte. On an average, the lifespan is around 6 to 16 days and they are found in the connective tissues or at other sites of possible pathogenic challenge such as gastrointestinal tract, the lungs and in organs like liver and spleen. These macrophages also exhibit phagocytosis. Coming to the functions of the macrophages, these macrophages, they ingest and kill the microbes. To ingest and clear the dead cells and unused cells, macrophages play an important role. They secrete cytokines upon activation. After the activation of the macrophages, cytokines will be produced and they activate the many lymphocytes. These macrophages serve as antigen presenting cells to display the antigens to the T lymphocyte along with a MHC molecule. They also help in the angiogenesis, that is in formation of blood vessels. So these are the few listed functions of macrophages. Next we see about the mast cells. These are derived from bone marrow cells and contain histamine and other chemical mediators of allergic diseases. These mast cells are involved in allergy reactions or hypersensitive reaction. The mast cells express the receptors for immunoglobulin like IgE and IgG antibodies. They also provide the defense against helminth infection. Coming to the basophils, they are structurally and functionally similar to the mast cells and mediate allergic conditions. These are granulocytes. The granules of the basophils contain acidic proteins which bind to basic dye, that is hematoxylin. Next is eosinophils. They are granulocytes present in the blood, contains the enzyme required to damage the cell wall of parasite. They act on the cell wall of the parasite. The granules of the eosinophils contain basic proteins which bind to acidic dye, that is eosin. Next is dendritic cell. These dendritic cells are specialized antigen presenting cells which captures the microbes and microbial antigen, process it and transport them to the lymphoid tissues to be recognized by lymphocytes. So they help in making the immune system to easily identify the pathogen. It will present the antigen molecule, or it will process the antigen and it will present the certain molecules on its surface, so that immune cells and immune system can easily recognize the pathogen and activate the immune reaction. They activate the naive T cells. Naive T cells means unchallenged or new T cells, and form a bridge between innate and adaptive immune response. In earlier videos, we know whenever there is infection in the body, both innate and adaptive immunity act together to clear the infection. So here the dendritic cell play an important role in intercalating innate and adaptive immune response. They are widely distributed into many organs and epithelial surface. These dendritic cells are present in many organs and epithelial surface. There are different types of dendritic cells. Among them, the plasmacytoid dendritic cells are the subpopulation of the dendritic cells. These are the type of the dendritic cells involved in recognition of the virus infected cells. Involved in recognition of virus infection inside the body. Naive lymphocytes. The lymphocytes that are not previously encountered with the antigens, that are not challenged with the foreign substance, are called as naive lymphocytes. They trigger the adaptive immune response after encountering with the antigen. These are the newly formed lymphocytes not challenged with any of the foreign substances or any of the antigenic derived molecules. Lymphocytes. These cells are derived from the lymphoid progenitor cell. These are the cells of the adaptive immune system. There are two subsets of lymphocyte. The two major subsets of lymphocytes are CD4 receptor expressing lymphocyte, CD4 receptor expressing CD4 plus or CD4 receptor displaying lymphocyte. They are called B lymphocyte. And another one is CD8 receptor displaying lymphocyte. We call them as T lymphocyte. You will be learning about these T lymphocyte and the B lymphocyte in the coming video in detail. B lymphocytes involved in the production of antibodies. These are basophils derived lymphocyte. These are called basophils derived lymphocyte, and they are involved in production of antibodies. Whereas T lymphocytes involved in the production of cellular immune response. They are involved in cytotoxicity, cellular immune response. So array of cells will be involved in killing the pathogen or in eliminating the infection. These are thymus derived lymphocytes. These are thymus derived lymphocytes. Next, another cell that is developed from the lymphoid progenitor cell is the natural killer cells. These are the lymphocytes in the same family as T and B lymphocytes, coming from a common lymphoid progenitor cell. However, as the cells of the innate immune system, the natural killer cells respond quickly to wide variety of pathological challenges. Apart from the innate immune system, these natural killer cells respond quickly, instantaneously to wide variety of the infection. Natural killer cells are best known for killing virally infected cells and detecting and controlling early signs of cancer. These are involved in killing the virus infected cells, and they are also involved in detecting and controlling the early tumor development. Specialized natural killer cells are also found in the body, especially in the placenta, and may play an important role in pregnancy. Effector and memory lymphocytes. So these are lymphocytes. They circulate through the normal bloodstream and are responsible for systemic immunity against the particular pathogen. The memory cells are important for providing protection against the second exposure of the antigen. When the pathogen enters inside the body, naive T lymphocytes will be challenged in a population of the T lymphocyte or in the population of lymphocyte, and they start producing the respective effector mechanism, and they finally clear off the pathogen. And these naive cell, they undergo repeated clonal expansion, they undergo repeated cell division, and all these daughter cells, all these newly formed the lymphocyte against that specific pathogen infection will specifically recognizes only that pathogen, and they clear off the pathogen, and the part of this population will retain that memory, will remember that pathogen for the further encounter or for further infection. They are called memory cells. They are called memory cells. So these memory cells are important for providing protection against second exposure of the antigens. And these memory cells are produced in the body during their first encounter with the antigen and expand. Expand means they increase their population, they undergo clonal expansion. That you will be learning about clonal expansion and immunological memory. They expand, they increase their number, and following their repeated exposure. So these are effector and memory lymphocytes. I hope you understood the cellular basis of immune system. This is very, very important before we move on to the further aspects of the immunology, like adaptive or the acquired immune reaction. In that you will be finding all these cells, their role, and you will be listening about these terminologies very frequently when we start learning about the acquired immunity in detail. Thank you.